ሰላም ጤና ይስጥልኝ ውድ ኢትዮጵያውያን ኤርትራውያን ወገኖቼ ዛሬ ቀኔ በጣም ቢዚ የሚሆንበት ቀን ነው። ለምን በጣም ብዙ አክቲቪቲዎች አሉ። እንግዲህ ጠዋት ልጄን ወደ አሜሪካ ለማትስ ኮምፒቲሽን ሶስተኛው ልጄን እልካለሁ። ከዛ ደግሞ ባለፈው ቪዲዮ ላይ እንደነገርኳችሁ ዶክተር ሙሉ ገለቱ ያዘጋጀችውን ፕሮግራም ማየት አለብን። ሄጄ ሰፖርት ማድረግ አለብኝ። አንፎርቹኔትሊ ሙሉ ቀን መዋል አልችልም። ነገር ግን የተወሰነ ሰዓቶችን እሷን አይቼ እመጣለሁ ብያለሁ እና ያው በጠዋቱ ተነስቼ ስለባብስ ቁርስ ስበላ ልጆቹ ቁርስ ሳስር ምናምን በጣም እንዳይረዝምባችሁ ብዬ አልቀረጽኩትም። ሬዲ ሆኜ ልጄን እንግዲህ ልሸኝ ነው እሱ ወደሚሄድበት ኮምፒቲሽን እና ከዶክተር ሙሉ ፕሮግራም በኋላ ደግሞ ሌላኛው ልጄ ከዋተርሉ ይመጣና እሱ ደግሞ ዛሬ የሮቦቲክስ ቲም ኮምፒቲሽን አለን ነገና ከነገ ወዲያ ኦሽዋ። ስለዚህ እሱ ደግሞ አሁን በፕሮግራሙ ውስጥ ባይኖርበትም መጥቶ ግን ጊቭ ባክ ቱ ዘ ኮሚኒቲ ያደርጋል። ኮች ያደርጋቸዋል፣ የሚነዱትን ልጆች ያስተምራቸዋል። ኮምፒቲሽን ላይም መንዳት አይችልም፣ ሮቦቱን መንካት አይችልም እንጂ ቢሃይንድ ዘ ሲን ልጆቹን መርዳት ይችላል። ኮች አላቸው እያንዳንዳቸው። ያው ነገ ከነገ ወዲያ ማሳያችሁ ይሆናል። ስለዚህ የዛሬ ቀን ውሎዬ ይህንን ይመስላል። ታዲያ በዚህ አጋጣሚ በጣም ክራውድድ በሆነ ፕሮግራም ላይ ከጠዋት ስንነሳ አንዳንዴ አስተውላችሁ ከሆነ ልጆቻችን በቃ ይሄ አልተሳካም፣ ይሄ ነገር ማታ ፓክ አድርጋችሁ አስተካክላችሁ እንኳ አንዳንድ ጉድለቶች አይጠፉም። ስለዚህ ልጆቻችሁ መንገድ ሊወጡ ሲሉ በተለይ ውድድር ቦታ ሊሄዱ ሲሉ እናንተ ደግሞ ቀናችሁ እንደዛሬ ቢዚ ሊሆን ሲል አደራችሁን ትንንሽ መሰናክሎች እንዳያበሳጯችሁ፣ ቀናችሁ ሰላም እንዲውል። አንዳንድ ጊዜ ለነሱ ደክመን ግን ሰላም ካልሰጠናቸው ልጆቹ ደስተኛ ሆነው አይውሉም ብዬ እላለሁ። ይሄ እንግዲህ 10ኛ ክፍሉ ልጄ ነው፣ ማቲው ሮንዳሳ ይባላል። ማቲዬ ዛሬ ፔንሲልቬኒያ ዩኒቨርሲቲ ለማትስ ኮምፒቲሽን ይሄዳል። ይሄው አሁን ልጄን እዚህ ከተማሪዎቹ ጋር አገናኘውና አብረው በትምህርት ቤት ነው የሚሄዱት። ከዛ እኔ ደግሞ ያው እንደነገርኳችሁ ወደ ሌላ ፕሮግራም እቀጥላለሁ። ማቲዬ I'm so proud of you and I wish you the best competition and may God help you and protect you on your way. I love you. Bye bye. እንግዲህ ኦኬ መንገዳችን ጀምረናል። እንግዲህ እንቀጥላለን ማለት ነው። መልካም ቀን። ወደ ሚሲሳጋ ካምፓስ ማነው ዶክተር ሙሉ እና ሌሎቹ ያዘጋጁት ፕሮግራም ጋር እየመጣሁ ነው። ይሄ ሚሲሳጋ ዩኒቨርሲቲ ኦፍ ቶሮንቶ ካምፓሱ ነው። እዚህ ቢልዲንግ ላይ ነው፣ 3ኛ ፍሎር ላይ ላብ ያላት እና የተለያዩ አበሾችንም የጥቁር ልጆችንም ነው የምታሳትፈው። በእውነት በጣም ሰፖርት ልትደረግ የሚገባት። እኔ ለምሳሌ ዛሬ ትልቋ ልጄ ዩዥዋሊ የእሷ ፕሮግራም ላይ ቮለንተር ታደርጋለች። በት አንፎርቹኔትሊ ዛሬ ፕሮጀክት ስላላት አልመጣችም። ፕሮግራሞች ሲገኙ ፕሮግራሞችን መከታተል አለብን። ይሄ ቀላል ነው። ጠዋት ተነስቶ ይህን ሁሉ ልጅ አዘጋጅቶ ምናምን አይደለም፣ ግን መሰጠት ግዴታ ምለው ይሄ ላይ ነው እና እስቲ ተከታተሉ። ፕሮግራሙ ምን እንደሚመስል አሳያችኋለሁ ዛሬ። ይሄን ይመስላል ካምፓሱ ማለት ነው። Hi, sorry, I want to go to 3rd floor. So should I take elevator or?
You can take the elevator or the stairs. These will take you to the 2nd floor, and then you can take the elevator up one more.
Okay, thank you. Thanks. Okay.
It's growing. It is 37 degrees centigrade. It's mimic our body temperature, and also 5% CO2. Because O2 is overall here, and then it compensates 5% CO2 and O2 to mimic our body temperature to grow the cells. We are growing different kinds of cells, different cancer cells or normal cells to test the drug. Most of the time we grow lung cancer cells, leukemia, blood cancer cells, breast cancer cells, cervical cancer cells, whatever cancer you want, we grow and test the drug. And these are the containers that we are storing our cells. It is liquid nitrogen. You can keep the cells in liquid nitrogen whatever you want. 20, 30 years, nothing will happen. You have to refill the liquid nitrogen. So freeze them. Whenever you want, you just take out and then grow the cells. So this is the storage place that we are storing our cells here. See, the liquid nitrogen. It's very, very cold. Minus 180 degrees centigrade. Minus. Very cold. So these are the incubators. I have several cells here I can show you today under microscope. It looks like this. It is closed, humidified, and in different container and different media to feed the cells. We have media to feed the cells, otherwise they will die. That is, we have to add vitamins, minerals, and serums and all this kind of thing to feed the cells, to grow them, to divide them, and so on. So I can show you some cells today. These cells called HeLa cells. Have you ever had HeLa cells? This cells is cervical cancer cells. It derived from a black woman, the first cell line in research lab 50 something years ago. Her name is Henrietta Lacks, and they took her first name and last name spelling to make the name of the cell, HeLa. And she was diagnosed with cervical cancer, and then they took the sample and then they took to the lab this cell and grow. These cells is crazy growing cells, and they started growing them because most of the time when you take human sample directly, it is very hard to grow. You have to keep them, looking them, adding media, adding nutrients to grow them and to make cell line. This is the first cell line in research, and it distributed all over the world to do research, to develop vaccine, to develop drugs. And Henrietta Lacks, they took her sample without consent. So during that time, there is no consent. One guy took the sample and then start researching. But she didn't know, and she was died. And her family, after a long time, they just go to the court or something, and then they compensated for that. So there is a very fascinating book called about Henrietta Lacks. Maybe you can find it and then read it. Very interesting. How they developed this cell line, how they took it, how she suffered from cancer, and how she died, all this kind of, the history of her and the history of her cell lines. So there is a book about that. So I can show you the cells. It is growing now. Yeah, you can come have a look. Each of you. 1 second, I will give you gloves. Sorry. Small or extra small. You can take some gloves to focus. Small or extra small. Just use this one to focus. Use this one because everybody's eyes is different, so you have to focus to see something there. There is something there. What is there? Yeah, they are adherent cells. They stuck to the container, the flask, and they grow and they become dense. You know, they are not individual cells. They are touching each other and then growing. Next. I want to see. long do they live outside the incubation? If you keep overnight, they will die outside the incubator. So couple of hours it's okay, but overnight you can't. You focus, if you cannot see, you just use this one to focus for your eyes. See, did you see? Yeah. Use both eyes open and use the binocular both eyes. Don't close your eyes and then just check. If you cannot see, just adjust the focus on these eyes. Okay. Hey, ciao.
Wait,
You want to
I'll
use?
come back later.
Okay. Manu Maya, just open both eyes and focus with this. These are cervical cancer symptoms. So we are screening the drug using these lines. And you want to see?
You see some white stuff?
Taisha? Yeah, full of the flask. Okay. Okay. The next one is leukemia cell line. It's called CML, chronic myeloid leukemia. We have two different leukemia. Leukemia is a very broad cancer, a blood cancer. There is AML, there is CML, there are FPL, different kind of leukemia. So I'm showing the chronic lymphocytic leukemia, blood cancer. Yeah. You can see this one easily. Come and see. Because it's blood, yeah? Blood is a fluid, yeah? The cells also not attaching the surface. But breast or cervical is a solid tumor, yeah? They are tumor. So they are adherent potency. So they are adherent to the flask or the plates. But blood cancer is just floating like blood. Go ahead. If you have question, just ask any question. This is blood cancer. It's very tiny circle cells. Huh? The other one is flat and touch the surface and stuck to the surface. That is adherent cells. The cervical or breast or lung, they are tumors, solid, so they are low potency. There are very few. I just split yesterday, so there are few now. They grow. If you keep them 2 or 3 days, they will be full of the flask. So they divide and then make more.
So you're doing this, Moulier, is to see how fast they grow?
No, I went testing the drug. The chemist making the drug
drugs,
and then see the potency of the drug. I dose them to the cells and then how potent this drug killing these cancer cells.
Okay.
I have normal cells also. I just dose the normal cells. If it is killed, this drug is toxic. It's not drug. So I will compare the normal cells versus the cancer cells. So I will dose them and see the potency of the drug. The chemist producing the compound, the drug, the biology is the cancer cells. That is a good thing. But if it kills the cancer and the normal cells, that is not good. It's toxicity. We are testing also toxicity. So if it kills both, we just trash the drug. So they will make another one.
How do you know how much dose you have to use?
We optimize the drug. We just dose different concentration and find out which one is the optimal concentration to kill the cancer cells, but not to kill the normal cells. Yeah. And I don't have the cells. I can show another one. We also working with not cancer, rare diseases, genetic disorder diseases. It's called DMD, Duchenne muscular dystrophy. And on this,
It's hiding here.
hi, where Duchenne muscular dystrophy is a muscle disease. Most of the time, boys are affected because the mother's gene is contained, this mutated gene. She's passing to the son because the chromosome the mother has. Oh, sorry. We have X and Y chromosome. Yeah, the boys are getting Y chromosome from the father and X chromosome from the mother. And if he got the mutated gene X chromosome from the mother, this boy will be sick with DMD. So they diagnosed when the boys are 4 or 5 years old, and they become deteriorated when they are growing up because the muscle regeneration is stopped. When they are teenagers, they be on wheelchair, and the life expectancy of these boys will be 20-25 years because they will die with a heart failure or something because the muscle is not working and deteriorating. It is degenerate the muscle. So we are looking at drug that treats this kind of diseases. So I can show you. I don't have live cells, but I make a picture of the cells that when I did this experiment, I can show you.
So they're getting the disease from their father.
Their mother.
If it's their mother,
Yeah.
they're taking the X chromosome, so there's higher chance of females, right?
Yeah. If they got the good X chromosome, they do 50%.
So it's more higher in the woman.
Yeah, this is in woman X chromosome mutation.
Yeah.
So they will get this mutated gene from mom, and if she pass the normal gene, normal X chromosome, they will be okay. If she pass this gene to her girl, she will be okay because she gets X chromosomes from the dad, normal, and she get mutated chromosome from mom, and she will be recessive. But this girl will pass to their son. Only boys are affected with these diseases.
But it's going to be higher in girls since the girls are taking the X chromosome, and it would have been more if it was on the Y chromosome, right?
No, it's not Y chromosome. It's X chromosome in the dad. In the mom.
I think it's because the women are the carrier
Yeah.
and the passing, but the women are not the affected one. You know,
The woman is a recessive. They have recessive gene, you know.
So, yeah, I think
And the woman is
it's XY, not the girls getting rather than the boys. But you carry the gene passing the boys.
yeah. This is we did culturing this on plates and making muscle on the plate. We differentiate using the drug. Yeah, you can see. These are the cells that we plated on the petri dishes, and then we treated with the drug and without the drug. So when there is no drug, they are not differentiating much. But when you put some drug, see how they differentiate muscle-like cells. You see, they elongated, multinucleated, and all these kind of things. You see, we tested several drugs, and you can see, yeah. For example, these drugs are produced in our lab, and this is a control, and this is negative control. See, in the negative control, you cannot see this fiber, muscle fiber. There is no differentiation. But on these drug-treated cells, there is differentiation. You can see the muscle fibers, and they are multinucleated because muscles have energy. To get energy, the cells be not one nucleus, but multinucleated. So you see, they are multinucleated nucleus. So we are trying to find a cure for this disease. There is no cure so far, but we are screening different kind of drugs to treat this young boy because it's very devastating for family. They will find out when the boys are 4 or 5 years. So this project started because one of our colleagues has a son who diagnosed these diseases, and he fundraised by biking from Hamilton to Ottawa and donated to this lab to do research, the money. So because of that, we started this project.
So there is no way that to prevent before to passing the gene to.
So far, there is no way because how do you know? The woman also don't know. Yeah, don't know if she has this mutated gene. They will find out when they have a boy, you know.
But they can have genetic mutation check-up, right?
This time, they can do this genetic mutation before they have a baby, kids or something. The technology is growing up now. It's good now to test what mutation you have or something. But money is very important, you know. You have to have money to do all these kind of things. So the research also developing now, and we are trying to find a cure for this disease also, parallel to the cancer treatment. Anybody of you have question? Any question? Okay. So you will go to other portion of the lab and see how chemists making drug and how biophysicists doing these proteins and all these kind of things. They can show you. This is a cell biology. If you have any question, just let me know. Ask me anytime. Okay.
guys are very fortunate to have this opportunity because no easy to access in the lab in today's day. So if you say thank you, Dr. Mulu. Dr. Mulu.
No problem.
So much.
Have rest. I have. Yeah.
How many groups are there?
It was 4, but now it is 5. Yeah.
We do medicinal chemistry here. Do we know what medicinal chemistry is?
Confusing terms, but essentially neuropathy is diseases that cause nerve damage, and this can be due to a variety of reasons. So chemotherapy actually causes nerve damage in the peripheries. So cancer patients often also experience neuropathy. So yeah, but what we do here in this lab is we study of many diseases. So the gunning group historically does study cancers, and this is cancers of the blood and the brain. We do this by studying specific proteins in the body. Maybe Bilal has mentioned some of this, but we study specific proteins in the body that may lead to diseases. So for example, a specific protein that causes the cells to divide more rapidly and causing the cells to develop cancer cells, for example. So that's sort of what we do here. Have you guys visited chemistry yet, by any chance? Yeah? In one of the other groups? The very far in like this empty one. It looks empty, but there's like a little bit of clear liquid in there. Can you see it in there? In this ropey. Yeah, or like it's not empty. Yeah, so for example where my fingers are, there's like a little bit of clear liquid. That's actually a purified protein called albumin. And so what we did with the previous groups is we sort of turned it into this purpley color. So we do this by these two reagents. So this is called a BCA assay, but this is a specific type of acid. And then this reagent is copper sulfate, so it's like a really pretty blue color. And so if I were to combine these two together, what color would it turn into? Take a lot. Sorry? Purple? Okay. It's a good guess because you already saw some of the really nice. It's a good guess. And so this is copper sulfate solution. And what we do is we actually add this solution into those empty wells. And maybe we can get one person that would want to, since you have your gloves on, I'm going to call you up to demonstrate this for us. Right? So you're going to actually, this is putting your thumb on top. Right? And what happens is when you push down, so actually what we'll do is we'll take up the tip first. You're going to push down until you hit like a stop. Then you're going to go into the solution and let your thumb go back up. You see how it takes up solution? Mm-hmm. And then to release it, you're just going to push back down. Okay? Want to give that a try? You're going to go into the liquid. Push down into the liquid. Not up. And now you're going to actually put it right in between. Yeah, in the empty one, in between. Go a little down. There, now push all the way. Perfect. Alright. Maybe I'll get like, you guys want to give it a try as well? Okay. So you're going to hold it like this. You're going to push down. Go into the liquid. Not all the way down, just into the liquid. And then here is you can slowly start to see, this is the very first one, right? It used to be green, right? It used to be like that. But now it's slowly turning into like a clear solution, and you can see a little tint of purple, right? Very little tint of purple. So this purple is going to get stronger. It's going to be actually somewhere in between these two. You see how this first one's a little bit lighter than that one? Yeah, that's because C actually has a little bit more protein, and you can see that through the color of the purple. So A had the least, B has a little bit more, and then C has even more than that. And so you can see slowly even the other wells where this one's a little bit green still, but this one's slowly starting to turn purple. So it's called a BCA assay. And so the more purple this solution becomes, the more protein there is. Right? And so visually, just by eye, you can tell if you have a good amount of protein versus not a good amount of protein. And but you can put this in.
A lot of beautiful girls here.
Deoxy because it doesn't have an oxygen like RNA. RNA has 2 hydroxyls, right? So DNA deoxyribo. Does anyone know?
Nucleic acid.
Yes, yeah. Deoxyribonucleic acid.
Very novel research and very
yeah, DNA is basically, do you know what it does?
Like I said, yeah, not really done before, but it's really cool. So what Tahir's going to show you is how you can mix 2 things in all of your organs, everything, all of your body.
Perfect. So it's basically the genetic code of life, right? It codes for all your genes, and those genes get turned into protein, and that protein builds up your skin. It builds up the skin color, your hair, the strength of your hair, the color of your hair, the eyes, your eye color. Like it just builds you up, right? That's protein. But that protein has to come from somewhere, and so DNA has all the information of life. So strawberries actually have a lot of DNA in them. And so today, have you guys ever done this before in high school? No? Okay, perfect. I'm so happy to do this.
እንግዲህ ውድ ወገኖቼ ምሳ ሰዓት ደርሶ አንፎርቹኔትሊ የዶክተር ሙሉ ገለቱን በጣም የሚያምር የላብራቶሪ ኤግዚቢሽን እንደ ኤግዚቢሽን ነው ያስደረገቻቸው ልጆቹን ቀን በግማሽ ነው ውዬ የመጣሁት። በጣም ዶቼ ኮምፒቲሽን አለ ኦሾዋ ቅዳሜና እሁድ። ስለዚህ ዛሬ ሄጄ ማደር አለብኝ እዛ እና ደግሞ ትልቁ ልጄ አምና ሚነዳ የነበረው ሮቦቲክስ ዘንድሮ ደግሞ ኮች ሆኖ ከትምህርት ቤቱ መጥቶ ለኮሚኒቲ መልሶ መስጠትን ስለሚያደርግ እሱ ልጄ ትንሿ ልጄ ወደ ኦሾዋ ስለማቀጥም ብዬ አልቻልኩም ፕሮግራሙ መጨረስ። በጣም አሜዚንግ ነበረ። ልጆቹን ጋውን አልብሳ ይቻላል ብላ መድረኩን አስከፍታልን። ሁሌም እንደምላችሁ ዛሬ አንድ ኢንፎርሜሽን ለመጠየቅ ሰዎች ያስፈልጓችኋል። እንደዚህ ደግሞ የላብ መድረክ አስከፍቶ ጋውን አልብሶ ለልጆች እንዲህ መሆን ይቻላል እንዲህ መስራት ይቻላል ብሎ የሚልን ሰው በፋይናንስም በፊዚካሊም ሰፖርት ብናረጋት ተጠቃሚዎች ምን ሆነው እኛ ነን። ዶክተር ሙሉ ገለቱ ኦልሬዲ የራሷ ስራ ያላት ሴት ነች፣ ግን እኛን ልርዳችሁ ብላ ስለተነሳች እንርዳት እላለሁ። ደግሞ ልጆቻችሁን ደስ ብሏችሁ ድሮፕ ያደረጋችሁም ሆነ የተሳተፋችሁ ሰዎች እናመሰግናለን። በዚህ ቀጥሉበት እላለሁ። እኔ በጣም ደስተኛ ነበርኩ። ኢትስ አሜዚንግ። ስለ ብዙ ነገሮች አስተማረቻቸው እና እግዚአብሔር አብዝቶ ይባርክሽ ደግሜ እንደምለው እና ዛሬ ምሳ ሰዓት ላይ አንፎርቹኔትሊ ጥዬ መጥቻለሁ። አሁን ደግሞ ለሜንቶር ነኝ እኔ እና ለሜንቶሮች ዛሬ ራት አበላለሁ ብዬ ቃል ገብቻለሁ እና አሁን የሰራሁትን ራት ደግሞ አዘጋጅና ወደ ኦሾዋ እሄዳለሁ። የኦሾዋ ጉዞዬንና ምን ይመስላል በድል ተጠናቀቀ ኮምፒቲሽኑ ወይንስ እንዴት ነው የሚለውን በሚቀጥለው ቪዲዮ እንገናኝ ይላለሁ። እግዚአብሔር አብዝቶ ይባርካችሁ። ይህንን ቪዲዮ ስታዩ ሼር፣ ሰብስክራይብ እና ኮሜንት ማድረጋችሁን አትርሱ። ሰላማችሁ ይብዛ። ወሰን ነኝ ከቶሮንቶ።
እንግዲህ አሁን ደግሞ ወደ ሮቦቲክስ ልንሄድ ነው፣ ወደ ኦሾዋ እንደነገርኳችሁ። ዛሬ ማታ ዲነር እኔ ነኝ ራት ማበላ ብዬ ሜንቶሮቹን ቃል ገብቻለሁ። ምክንያቱም ማታ ሮቦታችን ሴት አፕ አድርገን ለነገ ግጥሚያ የምንዘጋጅበት ጊዜ ስለሆነ ለቅዳሜና ለእሑድ እንኳን ምግብ ኦርደር አድርገናል። ይሄንን ሰላጣ ሰርቻለሁ። እንግዲህ ይሄን እንደዚህ አድርጌ ስጎ ሰርቻለሁ። ከዛ ደግሞ ይሄንን ስፓጌቲ ሰርቻለሁ። እንደዚህ አድርገን ራታችንን እንበላለን። እዛ ስንደርስ ደግሞ አሳያችኋለሁ።
እንግዲህ ይሄው እቃችንን ይዘን ወደ ኦሾዋ ለመሄድ እየተዘጋጀን ነው።
እቃችንን ይዘናል።
ኦሾዋ ልንሄድ ተነስቻለሁ። ከትንሿ ልጄ ጋር ነው የሚሄደው። ትልቁ ልጄ ያው እሱ ኮች ስለሆነ ቀድመው ይዘውት ሄደዋል። ስለዚህ እኔ ደግሞ ያው ትራፊክ ጠብቄ አሁን ወጥቻለሁ። ያው እንደነገርኳችሁ ትንሽ ራት ነገር ሰርቻለሁ። በጣም ቢዚ ነው የሚያደርገው። በተለይ ይሄ ወር ማርች ብሬክን አስመልክቶ ነው ብዙ ኮምፒቲሽኖች የሚካሄዱት። ብዙ ነገሮች ቀላል አይደሉም። ግን እኔ በጣም ደስ የሚለኝ ጊዜን ማሳልፍበት ቦታ እንደ እናት ተሰጥቻለሁ፣ አድርጌላቸዋለሁ። ነገ ከነገ ወዲያ በህይወት እንኳን ባልኖር እናታችን ለኛ እንደዚህ አይነት ነበረች እንዲሉና እኔ ደግሞ ኃላፊነቴን እንድወጣ የማደርገው ነው። እህቶች፣ ወንድሞች፣ በተለይ እህቶች በጣም የምነግራችሁ ነገር ምንም ነገር ዋጋ ሳያስከፍል ሪዘልት አያመጣም። ለልጆች ዋጋ መክፈል ቀላል አይደለም፣ ግን ደግሞ ውጤታቸው ስታዩ በጣም ደስ ይላቸዋል። እንደዚህ አይነት ኮምፒቲሽን ላይ ሲሄዱ ደግሞ ልጆቹ በጣም ኢንስፓየር ነው የሚሆኑት። ሁልጊዜ እንደምላችሁ የሚውሉበት ቦታ በጣም ኢምፖርታንት ነው እላለሁ። ወደ ወሸዋ ከ1 ሰዓት ተኩል ምናምን በኋላ እንደርሳለን። ሆቴላችን ስንደርስ ደግሞ እነግራችኋለሁ። ዛሬ ቀኑ እንግዲህ እየመሻሸ ነው። ቀናችን እንዲህ ነው ያለፈው ብዬ አስባለሁ።
ወደ ማታ ወደ 12 ሰዓት እየሆነ ነው። ይደክማል። ኮክም አድርጌያለሁ ትንሽ፣ ግን ደግሞ ደስ ይላል። እኔ ከልጆች በላይ ነው ኤክሳይትድ መሆነው ኮምፒቲሽን ምናምን ሲሄድ እና ልጆች ደግሞ ብቻቸውን ሳይሆን ቤተሰብ አብሯቸው ሲገኝ ሁሌ እንደምለው በጣም ሰክሰስፉል፣ በጣም ደስተኛ ነው የሚሆኑት። ለሁላችሁ ሪከመንድ አደርጋለሁ። ጊዜያችሁን ከልጆቻችሁ ጋር ስጡ። ልጆቻችን ውጤት እንዲያገኙ ከፈለግን እኛን ስራ አድካሚ ነው፣ በትብብር ነው የሚሆነው። እኔ ሁሉ ቤተሰብ ነው የሚተባበረው ለአንድ ልጅ ስኬት። ሁሉም አባቱም ልጆቹም ናቸው የሚተባበሩት። ስለዚህ እንግዲህ ወደ ወሸዋ
ኘሮግራም ኮሌጅ መጠናል። ከዚህ በኋላ ነው ሆቴል ምን ሄደው እነዚህ ባሶች የተወዳዳሪዎችን ነው ይዘው የሚመጡት። እኛ ደግሞ ይሄው ልጄ ትንሿና እኔ ሆነን እዚህ መጠናል። ወንዱ ልጃችንን ይዘን ደግሞ ወደ ሆቴላችን እንሄዳለን። የዛሬው ቀናችንን ይሄን ይመስላል። ይሄ ኘሮግራም ኮሌጅ ነው። እዚህ ነው እንግዲህ ነገ ውድድሩ የሚካሄደው። ዛሬ ሮቦታችንን ነው ሴት አፕ የምናደርገው።
ይሄ ቦታ ሮቦቶቹ ዛሬ ማታ ተስተካክለው ነው እዚህ የሚያድሩት። ነገ ነው እንግዲህ እያንዳንዱ ሮቦት የሚጋጠመው። ስለዚህ ለዛሬ ማታ ሮቦቶቹን እንደዚህ ቦታችን አስይዘን እናሳድራለን ማለት ነው። ከዛ ጠዋት ስንመጣ ውድድር በተወዳደርን ቁጥር ሮቦቱ ቢበላሽ የሆነ ነገር ቢሆን ምንድነው የምናደርገው? እየመጣን እንሰራዋለን ማለት ነው። እንደዛ ነው የሚሆነው ማለት እዚህ ኢንጂነሮች፣ ሜንቶሮች እዚህ ይቀመጡና ከዛ በኋላ ይላል ማለት ነው። የተለያየ ቲም ዋው look at this robot. Oh my goodness, I love it. Can I oh my god. Wow. Before you see this robot? No, go closer. No, put your eye glass. Go closer.
ድራይቨሮችን ኮች ለማድረግ እንግዲህ ሄዷል ወደ ሌላ ሩም ተሰብስበው ነው እንግዲህ የሚሰሩት ማለት ነው። ለነገ ጠዋት ነገ በጠዋቱ 8 o'clock ተነስተን ወደ ፍልሚያ እንሄዳለን። እርግጠኛ ነኝ እናሸንፋለን። እኛ የጥቁር ልጆች ደግሞ በቃ በደንብ አድርገን እናሳያቸዋለን። ኢንጂነሮች ነን፣ ጎበዞች ነን ብለን ወጠናል። እግዚአብሔር ደግሞ ሁል ጊዜ ከኛ ጋር ነው። ጌታ ይረዳናል። ችሞሮ እንግዲህ የሚካሄደውን ነገር አሳያችኋለሁ ወገኖቼ። ዛሬ አሁን 3 ሰዓት ተኩል ነው። ቀኔ እንደዚህ አለቀ።